Have you ever wondered if doctors could track cancer using nothing more than a simple blood test? It may sound futuristic, but today scientists can do exactly that using something called circulating tumor DNA, or ctDNA. To understand ctDNA, we first need to start with the basics. DNA. DNA is the instruction manual for every cell in your body. Inside each cell is a tiny library that contains the code telling that cell how to grow, repair itself, and perform its specific job. These instructions are written using 4 chemical letters, A, T, C, and G. Most of the time these instructions work perfectly, but sometimes the code changes. These changes are called mutations. When certain mutations occur, they can cause cells to behave abnormally. Instead of growing and dividing in a controlled way, mutated cells may start multiplying too quickly, refusing to die when they should, and ignoring the body's normal safety systems. That's what happens in cancer. Cancer cells carry mutated DNA that allows them to grow uncontrollably. But cancer cells don't live forever. As tumors grow, many cancer cells die and break apart. When they do, small fragments of their DNA spill into the bloodstream. These tiny pieces of genetic material are what scientists call circulating tumor DNA, or ctDNA. Think of it like this. Imagine a tumor leaving microscopic fingerprints in the blood. Each fragment carries the same mutations that were inside the cancer cell it came from. Even though these fragments are incredibly small, usually only about 150 to 200 DNA letters long, they contain valuable information. By analyzing these fragments, doctors can learn what mutations exist inside the tumor and how the cancer is behaving. To find these fragments, doctors use a test called a liquid biopsy. Unlike a traditional biopsy, which requires removing tissue from a tumor, with surgery or a needle, a liquid biopsy only requires a small sample of blood. Inside that blood sample are millions of pieces of DNA from normal cells. Hidden among them may be tiny pieces of ctDNA from cancer cells. Using powerful technologies like polymerase chain reaction and next-generation sequencing, scientists can scan the DNA and identify even a single mutated fragment among thousands of normal ones. This ability has opened a new window into cancer biology. Because ctDNA is constantly released by tumor cells, the amount of ctDNA in the blood can change over time. If ctDNA levels are dropping during treatment, it may mean the therapy is working and the tumor is shrinking. If ctDNA levels start rising again, it could be an early warning that the cancer is returning or developing resistance to treatment. In many cases, ctDNA can detect cancer activity months before it appears on scans. Doctors can also analyze ctDNA to identify specific genetic mutations, such as changes in genes like CRAS, TP53, or EGFR. Knowing these mutations helps physicians choose targeted therapies designed to block the exact pathways that cancer cells rely on. Researchers are already using ctDNA to monitor many types of cancer, including colon cancer, lung cancer, breast cancer, and leukemia. In some studies, ctDNA testing after surgery has been able to predict whether a patient's cancer is likely to return, allowing doctors to start treatment earlier and improve outcomes. What makes ctDNA so powerful is that it provides a real-time snapshot of what the cancer is doing inside the body, all from a simple blood sample. It doesn't cause cancer, and it doesn't help tumors grow. Instead, ctDNA acts like a message from the tumor itself, a tiny signal floating through the bloodstream, revealing clues about the cancer's identity, behavior, and future. As technology continues to advance, circulating tumor DNA may become one of the most important tools in modern cancer care, helping doctors detect disease earlier, monitor treatments more precisely, and guide truly personalized medicine. In a way, ctDNA is like hearing a whisper from the tumor, and in the fight against cancer, even the smallest clues can make a life-saving difference.